Good morning. Thank you. Thank you very much for that kind introduction. I'd like to also thank the organizers for the opportunity to speak today. I'm really happy to be here to share with you what we at Bluebird are doing in order to meet the challenges of bringing cell and gene therapy to patients. And I wanted to start with this slide. Making hope a reality for our patients is really at the center that everything that we do at Bluebird. And this concept of true blue, that really our patients are what guide us, is really important. We had a young man with sickle cell disease come and talk to us a few months ago. Young man, 18 years old, shared the story of his life where a lot of the key moments in his life have been spent in the hospital. He's had 2 strokes already. And you see a story like that and you think, we can do better. We can do better to serve those patients. And if you think about, some of the faces that are on this slide, if Darren or Ethan, if they had been able to live, what would their life have looked like? So that's really what drives us at Bluebird. And the other part of that is this concept of blue mojo. And it's that while we take what we do very seriously, we try not to take ourselves too seriously. And just to embarrass Margaret, who's in the audience here, we recently had our Halloween costume contest. There she is as the evil queen. I suggested that she wear this for all our face-to-face meeting with CMOs, but that wasn't so well received. So we at Bluebird have set very ambitious goals for ourselves. So by 2022, we want to have at least 2 products on the market and 2 programs nearing commercialization. And luckily for us, and as you'll hear, that does also bring some new challenges, we're really close to making that happen. So we just filed our MAA for lentiglobin and transfusion-dependent thalassemia, and we're on track to meet all of those objectives. And it's really been a journey that's been a long time coming. So the company was actually founded back in 1993. It was called Genetics at the time. And in 2009 and 2010, there was some pivotal data that was published in 2 diseases, adrenoleukodystrophy and transfusion-dependent beta thalassemia, that really brought a new influx of investment into the company, and that's when we were rebranded as Bluebird Bio. And since then, we've had a number of key partnerships that have enabled us along the way. We acquired some really important technologies, received some enabling regulatory designation, and then some of those key clinical trials that have brought us to the point where we're looking at commercializing our first product next year. And we continue to want to innovate across multiple fronts. So we at Bluebird are focused on 2 main disease areas, severe genetic diseases and oncology. And we have programs in each of those, and we don't do that alone. We have a number of great collaborators that really we're hoping to leverage our capabilities, our platforms, and then also find that right synergy with our partners that can help us bring these therapies to patient. So I'd like to tell you a little bit about the science of what we do and the platforms that underlie everything at Bluebird. So that's gene therapy, cancer immunotherapy, and gene editing. And I'll tell you a little bit more about each of them. But really underlying all of this is the fact that we're able to modify hematopoietic stem cells. And these cells are really powerful, and they can recreate all of the cells within the blood. And we take advantage of that in a number of our different programs, whether we're modifying these in our ALD or TDT programs, or whether we're then modifying T cells for our cancer immunotherapy programs. So we at Bluebird, when it comes to gene therapy, we use lentiviral vectors to deliver genetic information. So we use a modified form of HIV that's very good at getting DNA into the cell. And we do that through a transient transfection in a HEK293 cell line. So we transiently transfect with the plasmids that encode all the different parts of that virus, as well as their gene of interest. And then we harvest and purify that vector. Our next step is then to isolate target cells from the patient, so that can either be those hematopoietic stem cells if we're going after severe genetic disease, or leukapheresis to get patients' T cells in the case of cancer immunotherapy. And then we transduce those cells, so we genetically modify them outside of the body. And we grow sufficient cells to meet dose, and then the final step in the process is to test those cells and reinfuse them into the patient. So when it comes to cancer immunotherapy, in this case, we're not correcting a genetically deficient gene, but we're actually enabling T cells to be able to fight cancer. So we're inserting a chimeric antigen receptor that will be targeted to that tumor cell and also has that signaling domain that can recruit the immune system. And then finally, the third part of our platform is gene editing. This is a little bit newer to bluebird. So we, through an acquisition, use a technology for gene editing known as megatals. And really the way that we're thinking about this, and we'll be presenting at ASH this year some of our early preclinical data, is that we can use gene editing in combination with some of our CAR or TCR therapies that we're exploring to really go after some of those really difficult tumors. So especially in the solid tumor space, our thought is that if we can approach and attack the cancer from multiple different fronts, we hopefully can be very successful. So we're hoping that we can move this into the clinic shortly. So I'd like to spend a little bit of time telling you about some of the key diseases that we're pursuing at bluebird, and then I'll talk about some of the challenges that we're facing. The first is transfusion-dependent beta thalassemia. So this is an inherited blood disease that's caused from a mutation in the beta hemoglobin gene, and it requires frequent lifelong blood transfusions. And actually, diseases that result from mutations in hemoglobin are the most common inherited disease in the world. And it really has a high unmet need. So right now, the only treatment option is limited to allogeneic hematopoietic stem cell therapy, and typically that's limited to pediatric patients and only with a sibling donor match. And in addition to that, there's a number of severe treatment-related risks and complications. So one of the interesting things that we're able to do is the hemoglobin gene that we deliver has been mutated. It has a single point mutation that allows it to have anti-sickling properties, which you'll see is important later on. But what we're also able to do is that in a blood sample from our patients, we're able to distinguish how much of the hemoglobin that's there came from the gene that we inserted versus what's endogenous. So in this graph, what you want to look at is that pink bar, which is that hemoglobin that we've supplied through the lentiviral vector. So what I'm showing here is data from one of our clinical trials, and we're looking at hemoglobin concentration on the y-axis. And typical range is between 10 and 15 grams per deciliter. And what you can see is that 7 of the 8 patients, if you look at those pink bars, are producing almost 8 grams per deciliter of hemoglobin that comes from that lentiviral vector that we delivered. And in addition to that, 7 of those 8 patients are now transfusion-free at the 6-month time point. So this is some really powerful data and is the basis of what we submitted in our regulatory application. The other disease that we're targeting is sickle cell disease. Again, this is caused by a mutation in the hemoglobin gene and results in sickling of blood cells. And this has, again, a really high unmet need. The median age of death for patients is in their 40s with this disease. And again, the only underlying treatment is that allogeneic hematopoietic stem cell transplant, again, with the same limitations that you have to have the sibling donor match, and there's a lot of complications that can result. And the story of our data in sickle cell, it went through a lot of ups and downs. So we treated the first patient for sickle cell and had fantastic results, and the patients actually didn't even need Advil anymore to manage the pain crises. And then the next 6 patients that we treated, we saw absolutely no clinical benefit. So as you can imagine, this caused a great amount of consternation in the company, and we knew, okay, we know that this can work, let's go back and figure out what's going on. And so it launched a big investigation, and what we found was there were a number of factors that hadn't been controlled and could be improved in the manufacturing process, whether that was the viability of the cells, the dose that was used, the percent of cells that were positive for vector. So the different groups that you're seeing in these charts are the first trial, Group A, and then Group B, where we made some improvements to that manufacturing process, and Group C, finally, where we really threw everything at it and really improved on every of the factors that we had identified. So again, what you want to look at in these graphs is that the height of that pink bar. So looking at the data from Group C, you can see that at 3 months, all of our patients are at above 30% for the anti-sickling hemoglobin that we supply. And then for the patient where we did have 6-month data, above 62% of that anti-sickling hemoglobin. So essentially, the patients are now asymptomatic. So again, some really powerful data that once we had figured out some of the manufacturing challenges along the way, we were able to apply that moving forward. The next disease I want to talk about is multiple myeloma, which is a lethal blood cancer that remains incurable. And one of the things about multiple myeloma, it has a target that we can go after using CAR T therapy, and that's BCMA, or B-cell maturation antigen. So we've designed a CAR that binds that target, and this is some of the clinical data that we've received. So the patients in these trials have had at least 7 other lines of therapies, in some cases as many as 20 lines of therapies before going on to this treatment. And if you look at the data that's highlighted in red there, you can see the objective response rate for these patients greater than 95%, and 50% of those with a complete response. So this is incredibly powerful data, and this is a program that we have in collaboration with Celgene. And really, as a result of this, we're pursuing, can we look at this therapy, BB2121, in earlier lines of therapy to be able to help more patients? So we do have a planned phase 3 trial where we'll be applying this as a 3rd-line therapy. And then we're continuing to explore options. Can we pursue this as a 2nd-line treatment and, potentially even as a 1st-line treatment? And then finally, the 4th and final disease that I want to touch on is cerebral adrenoleukodystrophy. And I think the story about ALD really exemplifies what bluebird is all about. This is a very severe, often fatal neurological disease that manifests in boys. And as our CEO said, this is a genetic death sentence. And as a parent, this is your worst nightmare. It's a very rare disease, but if it's your child that's affected, this is not rare to you, right? And there's no other options here other than, again, the allogeneic hematopoietic stem cell transplant and all the risks that are associated with that. And again, what we've done here is, using our lentiviral vector platform, have delivered a copy of the gene that's deficient in this disease. And what you can see in this data here is that, so I'm no expert at reading scans, but I've been told white is bad. So if you look at the panel all the way on the right, that's an untreated patient. And what we see with the patients that were treated with our therapy is that we can completely halt the progression of this disease. And this is really powerful data that, again, speaks to, what's the potential that we can give these kids now. So you can see from this data that the patients were free of major functional disabilities once they'd received this treatment. So this is great. It's a great place to be in, to have all of this positive clinical data. But what it presents is obviously a supply challenge for us now, right? We have to be able to supply our drugs to patients in an uninterrupted way. And so how do we scale that, and how do we approach that challenge? So the first part is that there continues to be, as we heard before, certainly limitations on how much global vector manufacturing capacity there is out there. And we at Bluebird have certainly exploited a lot of that capacity. The other challenge is that there is an initial wave of therapies that are transitioning from clinical to commercial, which continues to stress the capacity in the system. And then finally, this is a very active field, and as many as 39 gene therapies could be approved by 2022. So how do we, at Bluebird, approach this path of how are we going to supply our patients with our product? And we've approached that from multiple angles. The first is that we have expanded our manufacturing footprint, so we have brought on more partners that we can manufacture vector with. And then the second decision that was made back in 2017 was that it made a lot of sense now to internalize some of that vector manufacturing capacity so that we could complement what was in the network of CMOs that we have. And then finally, not only scaling out, but also, as the previous speaker mentioned, scaling up and really finding process improvements is going to be key in order to be able to supply more material. So we have been working on a suspension platform as that next generation manufacturing platform, which can get us a lot more out of our manufacturing runs. So when we were looking at selecting our site for our internal vector manufacturing plant, we considered a number of factors which really forced us to look at some of the biotech clusters around the country. But really key to our final selection was our access to talent. You know, this is such an industry where this type of CMC talent is in high demand, and so we really wanted to be able to have great folks come to work with us, and that's why we finally made the decision to locate the plant in the Research Triangle area in North Carolina. And so we were able to acquire a building back in 2017, and we're now actively working to complete construction and fit out of that building so that we can start manufacturing as soon as possible. And we designed that facility to be able to produce any product, so we wanted to be able to supply both clinical manufacturing and commercial manufacturing from that facility. It can pose certain problems, obviously, if you have a commercial facility that you're also using for clinical, right? You're opening yourself up to a lot of scrutiny, but it was important for us to have that flexibility as we bring in new products through the pipeline. And then we also wanted to design the facility to be able to support any platform. So while we have the adherent platform, we also have this next generation suspension platform. So we wanted to make sure that our facility is able to support manufacturing of that platform, and then designed a flexible layout that allows us to accomplish all of these objectives. And you know, the decision to internalize manufacturing when you're a clinical phase company is certainly a challenging one, because internalizing that manufacture brings a number of challenges, right? There are a lot of activities which are first-time activities that you can leverage third parties with, to help you with, but that routine supply really requires that you execute at a high level consistently forever. And that requires much more than just buying a building, right? And it starts with building the right culture at that manufacturing plant. So we at Bluebird really pride ourselves on the culture that we've built, and we want to make sure that as we continue to expand around the globe, we don't lose what makes us Bluebird. So that was a very important criteria. Then bringing in those folks that have the startup in manufacturing expertise, but then also building those systems that are going to give you the predictable outcomes, right? So whether it's EH&S, quality systems, supply chain, business, and finance, all of these have to be in place to build that foundation to give you predictable outcomes out of that facility. And then the other part that I wanted to touch on is, as we transition from a clinical to a commercial company, there's really a mindset shift that has to happen, right? Because whereas we were focused previously on innovative therapies and bringing those to the clinic, now we have to think about how do we scale it, how do we deploy this within a commercial environment. And so there's a number of factors to consider as you look at, you know, what's going to change as we move from a clinical to a commercial company. And if we think about, for instance, the scope of our regulatory interactions, right? So up to this point, those regulatory interactions with global health authorities, we're asking advice on the things that we're about to do. But as we move to a commercial company, now we're talking about a lot of additional stakeholders that now have a vested interest in what you're doing, and they're bringing scrutiny to the things that you've already done. And whereas previously those regulatory interactions are very product-focused, now not only do they continue to be product-focused, but now they're interested in your processes and your systems and bring a lot more scrutiny to those as well. And then as we think about our day-to-day operations and what got us to this point, often it was a lot of really superhero-dependent efforts, right? People going the extra mile to get this done, to hit the milestones, never wanting to let a patient down. And that's great, but is that a sustainable model as you move to a commercial company? And so you have to think about, how do we become more dependent on our systems and have metrics that can drive actions so that we can get predictable outcomes along the way. And certainly we don't want to, you know, suddenly become big pharma, so it has to be done with some balance as well, right? And then the importance of great problem solving. A lot of the time as a clinical phase company, you're trying to fix what didn't work, and you have a lot of manual processes, whether that's on the manufacturing side or documentation, how we're capturing data from our processes. But what that can often result in is a really highly variable process, right? And now as we think about, okay, this is a mind shift, we have to supply our patients for the next 10, 20, 30 years with this therapy, what do we do moving forward? How do we shift that mindset? We have to shift from fixing what didn't work to permanently correcting it and preventing it at the root cause, right? And we also have to start thinking about how can we implement more automated processes? Obviously, being able to bring these therapies to patients all around the globe is going to require a dramatic reduction in the cost of goods, right? These are very expensive therapies, and we want to find ways where we can bring the cost down. So those efficiencies are really going to enable us to reduce the COGS. And so as we move from a highly variable process, we can first focus our efforts on stabilizing. How do we remove the variability? But then with these process improvements, how can we shift the mean and really continue to operate at a much higher level? And like I said, that's important for patient access, and that's important for ensuring reliable supply of our therapies. But I think that as we think about transitioning from a clinical to a commercial phase company, for us, the really important things are not changing, right? We want to stay true to who we are. We want to keep our core values. And most importantly, we want to make sure that there's one commitment that's never going to change as we transition from clinical to commercial, and that's our commitment to our patients. So with that, I'd like to thank you for your attention, and I'd be happy to take any of your questions.
Thank you. Yeah, floor is open for questions, so please step up to the mic. My question is, I love the slides and the sort of challenge of moving from clinical to commercial. A lot of people out here that you can set an example for. Sort of what has been the greatest challenge? I mean, you put a lot of thought into what you've done. What would you have done different, or what advice would you have for this audience or for someone going through that transition? What's sort of the number one thing or a couple things they should? I know that's a hard question, but
yeah, I mean, I think that having a leadership that really values the importance of the CMC operations, and you know, I think like that's been a struggle for me in past lives as well, is that CMC always seems to be an afterthought, but that this is really important, and especially in something like cell and gene therapy where the manufacturing process is so wedded to what we do, right? So the example that we had with sickle cell, where so much of it is go, 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 let's bring it to the clinic, but really we need to think about what are some of these attributes that are really going to influence our successful outcomes. And I think when you look at the draft regulatory guidances coming from the FDA, they're really forcing you to think, okay, let me think about how I'm going to characterize this product. What are my attributes? And let's start thinking now, you know, what are some of the process parameters that might influence those attributes? And to really start to have that mindset of building quality into our design, I think the earlier we can promote that. And that doesn't necessarily mean that we're doing these huge experiments to characterize all that, but starting to think about that from the beginning, I think really pays off dividends in the end.
Question?
Yeah, thanks for the talk. I just wondering if you could expand a little bit on the autologous cell part of the therapy. I mean, obviously, when you're getting those cells, they can be very variable depending on where you're getting them. And this is what we found, particularly even if you're going simple apheresis. Every clinical site does it a little bit differently. One may use a Fresenius device, another may use a Terumo, et cetera.
Yeah.
Is that important in the initial cell product? I mean, for us, it's very important what that initial cell product is. And if it is important, how do you decrease that variability? Are you going to try and take that in-house and have your own people try and collect the cells or manage that high degree of irreproducibility, and even in bone marrow collection, you know, one hematologist will do it one way, another will do it, you know, many different ways. Whether or not you do 2 sticks, 3 sticks, move the needle, and how much you draw at once, it all makes a very variable product, which I think is a very high degree of what the variability we're seeing in the autologous products coming in when we start the processing, which has a big impact on the final product.
Yeah, absolutely. And I mean, for our sickle cell trial, that was a key component that was found, was the variability, the way the collection was being done had a huge impact on how successful that therapy was. I think that We have within our company, the patient operations team and the apheresis team spends a lot of time thinking about how do we standardize that? How do we limit the number of collection sites, but so that at least then we're providing as much consistency as we can and working on these processes so that we're providing that standardization. I think the other piece though is there's going to be an inherent amount of variability that I think your process has to be able to withstand, right? I think, yes, you can make changes to how you're collecting and improve that, but your process has to be able to deal with that variability. And I think what we're investing a lot of resources in are tools to characterize that variability. And at the moment, it's let's just gather the data, right? We don't know yet what it's going to tell us, but we're using like very powerful different flow cytometry techniques or next-gen sequencing, or how can we look at this from every angle, characterize it, and then start to see what's going to predict, are we going to make dose for these patients? You know, how can our process be built to eliminate some of that variability?
So basically, you don't have the data yet to understand whether or not from a commercial aspect, you're going to need to limit to, you have to collect it this way, or we'll take all comers and we'll fix it later, I guess. There may, either one of those solutions is still, I guess,
I think it's still possible, but so I'm a cell culture person by training, and I just feel like that autologous, that drug product process to me is ripe with opportunities for cell culture scientists to go and characterize that. And again, I feel like there's, maybe you can eliminate some of the variability, or you can sub-select for a population you know is going to be successful, but if you can design your process to be able to absorb that variability and still allow you to meet dose, I think to me that's the dream.
If you have the cell of interest in the collection. And
correct.
The variability isn't coming from you not getting the right cell.
Yeah, and I mean, I know that we have made advancements in terms of how do we mobilize the cells at the collection point that really have had a lot of impact. And we've developed a second generation process where we have some transduction enhancers that really have helped a lot. So I don't want to minimize like the improvements that we've made along the way.
And that's almost a clinical trial in itself, because that's probably almost the most adverse events come from mobilizing those patients.
Yeah, right, exactly.
Thanks, that's a very nice talk, Ann. When you look at the, kind of related to David's question, the autologous cell processing and stuff, what technologies on the horizon do you think are going to have an impact? You know, there's this octane thing with, you know, GMP in a box and things like that. What do you see on the horizon that you think is pretty exciting?
Yeah, I think we have, you know, at Bluebird we have what we call our 11X challenge, which is a reference to Spinal Tap for any of those in the room that are wondering. But where, you know, as we're thinking about scaling from one patient, one process, one QC testing, right? And if you just multiply that by 10,000, that's not going to be sustainable. So we need to find new technologies. I think we're certainly looking at how can we automate it and bring that to the bedside as one possibility. And again, I think there's a lot of promise in how do we better characterize these cells so that we can select for the population that's going to be successful. I think the other tool that we have in terms of the gene editing that's hopefully going to become a powerful tool as we look to explore potentially allogeneic therapies, right, is now that we have the ability to edit genes, can we shift away from autologous and maybe get some of the same results without the immunogenicity as we're leveraging some of those gene editing tools?
Wonderful talk. I want to follow up on the last two questions as well. So there's some tools out there, I think, physical tools, but you also mentioned quality by design. Could you talk a little bit more about that type of strategy or other strategies that help you manage this type of heterogeneity for autologous products?
Yeah, I think that where we've applied a lot of those concepts is on the vector side, and then in our work with Aldevron on the plasmid side as well. So to think about what are the critical quality attributes of this process, and what are the parameters that might impact them, and then designing really robust studies around characterizing that. I know that, you know, on the autologous side, that presents a lot more challenges, right? Because you're doing your studies with healthy donor cells that's not necessarily representative of your final population. So I think that continues to be a challenge, and I think whatever analytical tools we can gain that allow us to get a lot more data out of a lot less cells are going to be really powerful in enabling that. But I think, you know what, I came from a biologics background and moved into cell and gene therapy, and I think those same concepts we're used to for antibodies, you know, characterizing our design space, and as more people from that field move into this field too. I think that helps to drive that mindset and convince people that, hey, I'm not just doing all these bunch of studies for no reason. Really, it's helping to drive robustness and consistency in the process.
Thank you.
Thank you.
Thank you. Maybe one final question. I think a lot of the presenters were asked yesterday, and you alluded to it several times, sort of where does bluebird sit right now in terms of location of cell processing? As you said, bedside, central, there's future technologies, but then there's the problems of today.
Yeah.
That bluebird has to deal with today, right? So sort of what is bluebird's strategy, or how does processing handle now?
Yeah, so right now on the drug product side, we've focused on having a few key CMOs that, whether it's geographically in the US or in the EU, where we're centralizing that drug product processing. I think certainly the bedside is part of our 11X effort. It was considered during our analysis, do we also want to internalize drug product processing? I didn't show our facility down in North Carolina is about 100,000 square foot, and we're only using about 50,000 of it, so we do have the possibility for expansion. But when we looked at what's most beneficial now, it was the vector side that made the most sense to internalize. So I think right now we focused on a few key partners in relevant geographical locations that we're focusing on. Yeah.
Okay. All right, thank you very much. Another round of applause, please.
Thank you.